Dear viewers, greetings. In this video, we are going to see about gel documentation system. This video contains the following contents. First, an introduction about gel documentation system, followed by principle of gel documentation system, types of gel documentation system, components of gel documentation system, procedure for gel documentation system, advanced systems in gel documentation system, applications of gel documentation system, advantages of gel documentation system, and finally limitations of gel documentation system. Gel documentation system. Gel documentation system is used in molecular biology labs for imaging and documentation of proteins which are suspended within polyacrylamide gel or agarose gels and nucleic acid. For DNA or RNA detection, the gel is stained with ethylene bromide, and for protein detection, the gel is stained with cohesive brilliant blue or silver staining. A gel documentation system is also called as gel doc, gel image system, or gel imager. Principle of gel documentation system. A fluorescent substance bound to nucleic acid is triggered by ultraviolet irradiation and causing it to emit fluorescent light. Ethylene bromide binds with the nucleic acid. The molecular weight and the concentration of the nucleic acid determines their bonding, which displaying a brighter shine if the molecular weight is large. However, if the molecule is smaller, the fluorescence will have a weaker shine. Types of gel documentation system. There are 7 types of gel documentation system. They are chemiluminescence, film autoradiography, laser, multiplex, Phospho Images, and finally CCD, that is charge coupled device or digital. The chemiluminescence is basically used for Western blotting. Film autoradiography is used for radioisotopes. Laser is used for multiplex fluorescence applications, highly sensitivity and precise quantification for radioisotopes. Multiplex is used to detect and image multiple fluorescent signals simultaneously, and this type of gel imaging system is also used to detect luminescent, colorimetric and radioisotopic signals. Phospho Images. Phospho Images is the advance of multiplex fluorescence applications and radioisotopes. It is more sensitive than multiplex gel imagers. It is more flexible in choosing emission filters. Finally, CCD, that is the charge coupled device or digital. Ethylene bromide, infrared, color fluorescence, tensometric, chemiluminescence and visible light detectors are available to take quantitative reads of nucleic acid, protein fragments, microplates and dot plots. Components of gel documentation system. There are 7 components of gel documentation system. They are imaging chamber, light source, blue light, camera, trans illuminator and finally software. The first component of the gel documentation system is imaging chamber. Imaging chamber is a light-proof chamber where the gel is placed for imaging. It prevents ambient light from interfering with the image capture process. The second component is light source. There are 2 light sources. One is from UV light and another one is from white light. UV light commonly used for viewing DNA or RNA which are stained with fluorescent dyes like ethylene bromide, which fluoresces under UV light. The white light is used for viewing the gels which are stained with colorimetric dyes like cohesive blue, which is used for the proteins. The third component of gel documentation system is blue light. Blue light is used for safer visualization of certain dyes like SYBR green or SYBR safe, which fluoresces under blue light. The fourth component is camera. A high resolution digital camera captures the images of the gel. It is typically a charged coupled device or CCD or CMOS camera that is sensitive enough to detect fluorescence or visible dyes. And the fifth component is transilluminator. Transilluminator is the platform on which the gel is placed. It provides either UV, visible or blue light illumination from below the gel for optimal visualization. The seventh component is software. The system is connected to a computer that runs software for image capture, editing and analysis. The software can enhance contrast, measure band intensity, calculate molecular weights and quantify the amount of DNA, RNA or protein in the each band. Procedure for gel documentation system. There are 4 steps in the gel documentation system. The first step is gel preparation. After electrophoresis, the gel is stained with a dye which is appropriate for the biomolecule of interest. For DNA, this could be ethylene bromide, while proteins are often stained with Kumasi brilliant blue or silver stain. The second step is image capture. The stained gel is placed on the transilluminator within the imaging chamber. Depending on the stain used, the appropriate light source like UV, blue or white is selected. The camera captures a digital image of the gel. The third step is image analysis. The captured image is processed through software to enhance clarity, contrast and sharpness. Quantitative analysis can be performed to measure the intensity of each band, which correlates with the amount of nucleic acid or protein present. The fourth and final step is documentation. The image can be saved in various formats, example JPEG, TIFF or PNG for record keeping, publication or further analysis. Advanced features in modern gel documentation system. The first advance is real-time imaging. Some systems offer the ability to monitor electrophoresis in the real time. The second advanced feature is automated band quantification. Advanced software can automatically detect and quantify bands, saving time on manual analysis. The third advanced feature is multi-channel imaging. Some system can detect multiple strains in a single gel by using different light sources like UV, visible or blue and filter sets. Applications of gel documentation system. There are 4 applications of gel documentation system. The first application is DNA or RNA visualization. Gel doc systems are primarily used in molecular biology to visualize DNA or RNA fragments which are separated by electrophoresis. This includes applications such as PCR product analysis, restriction digestion analysis, and gel purified DNA quantification. The second application is protein visualization. In proteomics, gel-doc systems are used to document the protein bands which are separated by SDS phage, isoelectric focusing or two-dimensional gel electrophoresis. The third application is quantitative analysis. The system can quantify the intensity of the bands, which allows for the measurement of the concentration of biomolecules in each sample. This is useful in applications such as gene expression studies and protein quantification. The fourth and final application is documentation for research and publication. GelDoc systems provides high quality images that can be included in research papers, presentations and lab reports. Advantages of gel documentation system. The first advantage is non-destructive. Gel documentation system do not interfere with the sample, allowing for further downstream applications such as gel extraction. The second advantage is quantitative data. Many systems offers quantitative analysis of bands, making it easier to measure the biomolecule concentration accurately. The third advantage is high sensitivity. GelDoc systems can detect very small amount of DNA, RNA or proteins when fluorescent or chemiluminescent stains are used. The fourth and final advantage is time efficiency. The gel documentation system provides quality and efficient documentation compared to the manual method like photography. Limitations of gel documentation system. There are two limitations. The first limitation is cost. Gel documentation system can be expensive, making them a significant investment for small labs. The second limitation is UV light exposure. Systems that use UV light can be harmful to DNA integrity if the gel is to be used for subsequent applications, and it also poses a health risk to users if proper protection is not used. Dear viewers, that's all about the gel documentation system. Thank you for your support. Thank you.